Hi, everybody. I'm Josh Steele. I'm a scientist here at Squirt, and I'm going to talk to you, sort of give you guys an overview of the San Diego River Investigative Order, where we are tasked to quantify human fecal loading in the lower San Diego River watershed. So sort of the background to this is there's a wet weather TNDL, bacterial TNDL in San Diego, and the compliance deadline is coming up very soon. And we've found from previous studies that not only is there human contamination throughout the San Diego River watershed, but there's also fecal indicator bacteria throughout it. And on top of that, the surfer health study that was done at the beaches at the mouth of the San Diego River showed that there was an increase in illness risk following wet weather. So not only do we find pathogens and human markers, we also find illness risk. And when I say that we find human markers and human pathogens, where do we find them in the lower watershed? Well, this is sort of a schematic representing the San Diego River. The main stem of the river is down the middle, and each of these little arrows off to the side are tributaries, and the red circles are human molecular source tracking marker HF183. And you can see that we find it pretty much everywhere we look. This is in wet weather during one storm, as an example. So this is sort of the background on what we're finding there and what we're trying to tackle.
How far is the ocean from there?
The ocean is here, basically, just not maybe a half mile from that point down there at the most, maybe even less. The cost of compliance is estimated in the billions of dollars, and the cost-benefit analysis was performed in San Diego, and they found that the most cost-effective way to reduce infectious illnesses, reduce public health risk, was to tackle human sources in the watershed. And you can see they looked at a number of different sources all along here, but the ones that have the biggest reduction are tackling human sources with high human sources, high and medium human sources, high, medium, and low, still dwarf anything else. And so that's sort of the basis for the investigative order itself, that this is the way forward to try and reduce public health risk. So what human sources are we talking about here? Like I showed you, we have the human marker that shows we have human contamination throughout, but that doesn't tell us anything about where that human contamination is coming from. So the main ones we've identified, and I'm going to talk to you about most of these in more detail throughout this presentation, are public sewer exfiltration and sanitary sewer overflows. There's a huge, it's a very large city of a few million people in the San Diego River watershed, so that's a major potential source. Private laterals in homes and businesses, not as well monitored as the public sewers, so it also could represent a very large source getting into the storm drains. On-site wastewater treatment systems or septic systems, not as widespread, but in parts of the county in the upper watershed, we definitely have areas that are mostly septic. And people experiencing homelessness in the watershed are another major source. And then finally, illicit connections, illegal discharges. I'm not going to go over that one because I'll sort of mention it briefly, but those are sort of scattered throughout. Okay, so how are we going to do this? The main goal of our work plan is we want to quantify the loading from all of these different sources throughout the San Diego River. And we're going to focus on, for the most part, on wet weather, because that's when we have the biggest health risk. We'll use these, use our loading estimates to try and provide a sense of what the relative contributions from these sources are. A way to prioritize what to tackle first. And then finally, we're going to try and identify factors that will lead to the greatest risk of human contamination. So when and where does that loading occur? And we'll do that all in the next 5 years. So to start with the very first one, we're going to look at public sewer exfiltration. We have two main approaches for doing this. The first one is a new technique that we're applying to look at sewer pipes and storm drains. And this is a technique that's been, and we're going to use the bacterial biofilms that are growing in the sewer pipes, which should be a unique environment compared to the storm drains, the bacterial biofilms growing in there, compare them, see if we can see that signal leaking into the storm drains. that's one approach. The second approach is we're going to actually go out and empirically measure the loss of volume in a section of sewer pipe in various states of repair, and I'll describe these in a little bit more detail. So for the biofilms, sewer pipes have a very different environment than storm drains, we hope. All of down here, down in San Diego, it's all separate sewer and storm drain. There's no combined sewer overflows. So we should expect that the sewer pipe has its own community growing in there, being fed by the sewage and all the other different situations down there, the anaerobic nature of it. Storm drains should be different flows, different things like that. The biofilm that's growing on these pipes is continuously sloughing off. And so what we can do then is we can sort of slurp up the biofilm off the sewer pipe and measure the entire bacterial community that's there and get a pattern that should be unique to the sewers. And then we'll do the same thing in the storm drain. So we're adapting. This is something that's been done successfully in Milwaukee and in the Midwest by a colleague of ours, and it hasn't really been tried out here. So ideally what we're looking for is these different patterns. So what you're seeing here, we have sewer samples on the bottom, sewer pipes and the storm pipe community on the top. The samples are going across, and up here we have different bacterial sequences, species. And you can see that we do find, there we go. We do find a pattern, a group of bacteria that are unique to the storm pipe that are not found in the sewer. It's great news. If we didn't find that, this wouldn't work very well. And we can find a group that are unique to both sewer pipes, that are found in both sewer pipes and not in the storm drain. Even better, we can find some that are unique to each sewer pipe. Now, we don't know, again, this is just preliminary samples. We haven't expanded this to a full set, but this is really encouraging that not only will we be able to tell the difference between storm pipes and sewers, we might even be able to tell differences in sewers or different areas. So far, this looks very promising. We'll have to see how it actually works when we expand it to more samples. Oh, come on. There we go. All right, so our second way of doing it is to do direct measurement of exfiltration. So what we have to do is isolate a section of sanitary sewer pipe and artificially create an increased flow like would happen in wet weather. And so we're going to do this using pumps, pumping water in, and flow meters to measure it, and then measure the volumetric loss over time. It's a little more complicated than just blocking it off and filling it up and watching it go down, but we think that this is a little more realistic. You'll have flows, you'll have things moving. It'll be more like what happens during a wet weather event in a sewer. Right now, we're designing a prototype, which will be a bench scale model. Once we have that up and running, just to see if we can get this to work with the pumps and the flow meters, we'll actually go out, and we've already identified a section in the upper San Diego River, lower San Diego River watershed, where we can go and actually block off the pipe and try it. So the way we're going to extrapolate this across the entire watershed is through a factorial design. And the factors, there's a lot of factors we could consider that were risk factors for the sewer pipes themselves. You can see here's just a subset of the ones that were identified: material construction, age, condition scores, groundwater height, soil type, depth of pipe to storm drain, et cetera, et cetera. We've basically focused on the first two to start, because we feel these are a proxy for some of the others. Age and material of construction should be a proxy for condition score, as well as some things like proximity to surface water, near it, how close it is to storm drains, things like that. We can always expand this, but in the interest of trying to get a reasonable extrapolation in a short amount of time, we're limiting it to the factorial designs quickly grow out of control. So that's how we're doing the public sewers. Next, I'm going to talk to you about how we're going to tackle private laterals. So there isn't a ton of data on private lateral inspections. It's not required most places, but one place it is required is in the city of El Cajon. And they have a requirement that you inspect your lateral, I believe, if you are remodeling or selling your home. So they've looked at 548 homes, and you can see that about a quarter of them have needed repair. So this is already a sign that this is something worth looking at. Most of the, and then, so this is of this quarter, you can see the ranked reasons for that: root intrusion, buildup, broken pipes, offsets. These all have different levels of leakage associated with them that could get into a storm drain. So our basic plan is to try to assess on a broader scale. We have that for El Cajon, that's great. Does that apply elsewhere? So what we'll have to do is actually do a survey of homeowners, provide incentives for them to let us come onto their property and inspect their laterals. We'll also use a similar direct exfiltration measurement strategy where we'll block the laterals and fill it with water. This one will be a little bit simpler because it's a shorter distance, and we can probably just measure the actual drop-in level. And we will use a stratified random design. So in this case, it would be, instead of looking at factors or age or anything like that, we'll try and get a random sampling across the watershed as much as we can, and then use that to give an estimate for the whole watershed. Okay, moving on to the on-site wastewater treatment systems, or septic. This can be a major problem. We weren't initially sure of this, but we've recently done a study in Los Coches Creek, which is one of the tributaries that was coming in, and other areas as well in the Lower San Diego watershed. And what you can see here, this is a measurement of a proportion of volume in this catchment. So you can sort of see different sub-catchments and then the tributary that goes into a creek. And the blue is water volume, and the orange is human marker. And so what you can see here is that while most of the volume is coming from a stormwater catchment that has sanitary sewers, nearly all of the human contamination is coming from a tiny little catchment that has septic systems. So this could potentially, in the areas where these are, be a very big problem. So how we're going to do this is we're going to basically try and be a little more selective than we were in that particular study, where we were looking at an area with multiple sources and try and find catchments that are pretty much exclusively septic. And if we can find them where there's no, where there aren't any people experiencing homelessness in the catchment as well, so much the better, where we can isolate that source. We'll sample these catchments during wet weather, where we can measure flow and we can measure human fecal contamination, similar to what you just saw. And then we'll be able to get an estimate of a per septic system average load. And we'll use that and the variability that we get from our different catchments that we are sampling to look across the entire watershed and extrapolate areas that have septic. Okay. For what it's worth, for the illegal discharges and illicit connections, we'll actually use a similar approach where we will target catchments where we believe they're happening and try and measure that or isolate that if possible. So our final one is estimating contributions from people experiencing homelessness. For this one, it's going to require a different type of analysis than we typically do. Initially, we're going to work with the folks at the Regional Task Force on the Homeless and the River Park Foundation and expand they have an annual census of the homeless population in the San Diego River watershed, and we're going to expand that to multiple times per year and just get a count of how many people are there and where they are. This will give us a sense of the scale. You know, if there is no one in the river that is contributing fecal contamination, it's probably not a big problem. If there are a few thousand people or hundreds of thousands of people there, it would be a much bigger problem. Even more importantly, we're going to look at their sanitary habits and take a survey of that. This would be, do they use facilities? Do they go to shelters at night? Are they only there part of the time during the day? This type of thing will tell us, to some extent, how much people are actually contributing fecal contamination to the river. Once we do that and get a sense of that, this will also feed into an upstream, downstream sampling where we can try and target areas where there is a high population and see if we see an increase in fecal contamination downstream of those population areas. And then finally, for the indirect deposit, where maybe there's people are defecating on the side of the river or somewhere where it only would come in once it rains, or in a watershed area like that rather than directly into the river, we're going to try and do some benchtop studies where we look at experiments, wash-off experiments, to see how long it persists, how long we can detect the signal, and how much actually gets in.
Are you also going to do like street or sidewalk experiments too with that, or is it just going to be on the banks?
For now, we've suggested we would do, I think, one that would be on concrete, and one that would be on more a softer substrate. So two situations at this point.
Okay.
Okay, so the current status of our work plan is that there's the draft work plan and quality assurance project plan are due today in, actually, well, hopefully they've been turned in, but they're due in 45 minutes. So, and we've, to produce this plan and go through it, we assembled a steering committee, which has the City of San Diego, the County of San Diego, the Region 9 Water Quality Control Board, the other named parties, the co-permittees in the Lower San Diego River watershed. We also have San Diego Coastkeeper and the San Diego River Park Foundation as members of the steering committee to represent the public and also the homeless population. In addition to that, we assembled a technical review committee with I think 6. Is that right, John? 6?
Yep.
Yeah, 6 national experts, including folks who developed the biofilm study analysis I've talked about, and folks from USGS, universities, CDC, places like that, so we can have independent review and oversight, and they've already given us suggestions on the work plan that's come in. Right now, the ball now has shifted to the regional board's court, and we'll see, they will give us their response and in the next 90 days, and we'll respond. And I have plenty of time to answer questions, so please feel free to ask whatever you want to know about our work plan.
Questions? Anybody? If you're online, if you'd like to ask a question, just unmute. I will try and check in the chat box.
So who's going to go on the river and talk to the people about their sanitary habits?
Well, the River Park Foundation already does conduct the annual census.
Yeah.
So we will sort of, I believe, have the same people try and conduct the more frequent censuses, but we're going to work with the Regional Task Force on the Homeless, as well as social scientists who specializes in homelessness, and to help design the survey, as well as train people to go out and talk to them. We haven't gotten to that step yet of choosing that social scientist. One of our technical review committees is an expert on urban populations and urban geography of the homeless. She's not going to be that scientist, but she's going to help us choose someone. Sure, something.
So this graphic, are they real data, or you just
No, that's real data.
Real data. Okay.
From a tributary into a catchment feeding into Los Coches Creek.
So for this pattern, okay, so this the orange bar is the H183.
HF183.
Mass.
Yes.
Or concentration.
That is mass in this case. So concentration and multiplied by volume.
Oh, that'll make the first catchment even more interesting, because such a small flow with such a
really high concentration.
Okay. The second question is, for the different septic catchment, it has a different distance with the terminus.
Mm-hmm.
are you at some point gonna actually consider the die-off of the species, including, you know, HF183 or virus or other things to help prioritize the drain shed or septic shed?
We are, maybe is the best answer I can give you. Not, I think that would be a step following first seeing what the relative contribution is compared to other sources. But no, it's an important thing to factor in for sure. I'm not sure if we would necessarily look at the die-off of HF183 or look for pathogens first. I'm not sure which would be our next step. But at this point, we are sort of assuming, you're right, that's not something that we currently have, that we're currently considering in this, and it could make a difference.
Yeah, but estimating risk, while it's a logical next step, it isn't part of this project.
Right. This is just, the order is to identify and quantify human fecal loading, but then the next step would be tracking, quantifying risk, things like that. So that's, we're trying to not jump ahead yet. Yes.
I have a question about that first septic catchment. First of all, I think the microbial community approach has a lot of promise, and we're excited about that. And you mentioned looking at the microbial communities in storm drain versus sewers and different types of sewers. Will that same approach be applied to the septic?
That would be, we would really like to apply it to the septic systems. It's a challenge to get those samples because usually it's private property, so someone would have to let you into their systems. What we will do is look in areas that are the septics and see if we can see a signal that appears to be a unique septic signal. But that'll be a mixed signal with the stormwater.
Right.
Necessarily.
Systems in parks.
Yeah, that would be a good place to start.
The other question I have about that high septic catchment is, do we know if there's recycled water or homeless communities in that catchment that could be providing signals as well?
We don't believe there are either in this particular catchment. It was a very small catchment, and to the best that we could tell, there were not.
Yeah.
No recycled water for sure, and then, yeah, we didn't see evidence of homeless encampments or homelessness.
So I really liked it's a really clever idea to like barcode each like sewer pipe or whatever with the microbial community. This is kind of a half-baked idea maybe, but have you thought about barcoding the pathogens too in each of those things? Is that possible?
It might be. You could do it for, it'd be trickier, or it'd be a lot harder to do with viral pathogens.
Okay.
To barcode them. You could do it with, there are some other genes you could do that might be better at barcoding the bacterial pathogens.
Okay.
So that is something that would be a logical next step. Yeah, it'd be a cool thing to investigate to see if it would work.
Yeah, and those are all just eDNA, right?
Yeah, yeah.
Great.
We had a question. We had a slide, and there were red circles, and they're asking if that was relative loads of concentrations.
Those were concentrations. Yeah, HF183 concentrations on the slide with red circles. I can go back to that maybe. No, I can't. Let me see.
Please help me touch it.
Yeah, so this was concentration, this slide in the river. So, yeah, so it is this giant, this really big circle with a very high concentration from a fairly small drain. So it's different than the septic one where we had low. This is just concentration to compare it to.
And those are samples at the outfall into the main channel?
They are usually just slightly upstream from the outfall itself. But where we, in an area that we could have a sampling, and these are composite concentrations.
Okay.
So over 6 hours, 12 hours, 12 hours.
So that one Marina Boulevard circle is quite large.
Mm-hmm.
When I've seen this slide presented before, it was indicated that it was discovered that there was an illicit sewer connection into a storm.
That is true, yeah. So that one was, they found, so the city of San Diego did investigate, and they found an illicit connection, I believe, from a fast food restaurant, as well as an area that was washing down sort of a lot of business that had vacuum trucks. And they were washing down their vacuum trucks, and the runoff was going into the storm drain. And so reducing, fixing the illicit connection and the discharge reduced that substantially in the next time we measured it. Yeah, that's absolutely right.
Do you know if there are plans to investigate some of the other big red dots for similar illicit connections?
I believe so, yeah. So the Los Coches Creek and Eucalyptus Hills, I know the county has been investigating. Let's see, the city has investigated others as well. San Diego State is looking at the sources on Alvarado Creek. Those are the ones I know off the top of my head. But yes, the basic answer is yes, there are. And I believe the Sycamore Creek, the Padre Dam is investigating that as well. And those are all everyone I mentioned is named in the IO and are also a part of it.
Pretty robust program.
It is a very robust program. Very exciting to be a part of.
But those of you online, if you have a question for Josh, you can unmute your phone and ask a question of him. I think we've satisfied everybody's curiosity.
One more question? Sure, we've got a few more minutes.
Okay. With regards to investigating exfiltration in the public sewer, you mentioned the factorial analysis.
Yes.
And I'd be curious as to how those pipe reaches would be selected. I understand there'll be some pipes in the Las Coches Creek. Right now, it's sort of to be done as sort of a proof of concept.
Yes, that's right. That's where we're.
And that's still moving forward with the county.
Yes, it is.
And but then secondly, how would the selections be made? You know, there's a very poor condition sewer pipes in the system. There's very good condition, average, and it would seem if you're trying to establish some sort of benchmark of potential leakage, you'd want the average.
So what we're going to do is we're going to try and cover age categories. So, you know, more than 50 years old or more than 75 years old, between 25 and 75 years old and newer than that. So that's one of the factors. So that'll be three different distinctions. The other factor then is pipe material are what we're focusing on. So clay, ABS, concrete, whatever the different pipe types are, that will ideally cover condition will be sort of folded into that as well. If we don't think that satisfies it, we can always add factors.
I would strongly suggest that condition has to be included in that because the condition of pipes don't necessarily correlate with age, especially with clay pipe, and it's very long life. Gasket material in clay pipe could be a factor for clay pipe prior to 65 before more waterproof gaskets were developed. The condition would play a huge factor.
Yeah, I mean that's why it's listed as one of our factors. For now, in discussing with the sanitation agencies, they thought age would be a better proxy at this point. So we're going with we're working with them, and if that doesn't work, we'll switch.
A question.
Yes.
So does a treated effluent have the human marker in it?
It can, yes.
It can, but it doesn't indicate that human marker or that bacteria is viable. It's picking up pieces of the DNA signature. So you can find HF-183 hits in disinfected effluent. It's not viable.
So it depends.
You can tell the difference?
It depends on the level of treatment, whether or not you find it. The HF-183 assay itself does not tell the difference, no. So in order to distinguish that, you'd have to know the water source, and you'd have to do additional investigation to either looking for pathogens, looking for activity. So you'd have to do something additionally to determine whether or not it's a risk. But like I said, we're not on that step yet. Right now, we're just trying to see where is it coming from.
When do we need to check back with you to find out how things went?
That's right. Well, so I mean, like I said, we just submitted the draft work plan, so we should hopefully start in early next year to actually start doing the investigation. So we will be able to provide updates as we go along, probably.
Yeah, I would think mid-year
we'll have some yeah.
Fantastic.
Yeah, and then probably we probably won't do much wet weather sampling this season. It would probably be
Eric, this is Rob. Can I jump in for the question?
Sure.
Or comment?
Yes.
Hi, for those that don't know me, this is Rob with the San Diego River Park Foundation. I'm on the committee for this effort, and we're excited to be involved. I just want to try to respond to a couple comments, if you will. We did look at the age of the pipes extensively. Some of the city of San Diego in particular has communities that are 80 to 100 plus years old. And so we do have still cast iron pipes, and there's known areas where we have some, shall we say, faulty private laterals. I think a lot of the effort was to first identify some of the known areas and kind of begin to work backwards. But clearly we fully understand that there's a lot of different things that can affect the condition of a pipe. Second of all, I did want to touch on homelessness because I was up at the previous meeting speaking about it. And, you know, the good news in the city of San Diego is, you know, the population of homeless is down 85 to 90% since our height in October of 2017 in the city of San Diego. But the city of Santee, which is upstream, another jurisdiction, has seen a tripling, quadrupling of the population in a similar amount of time. And so we know that the homeless population is very, shall we say, transportable as a potential source of some of this contamination. And it's one of the real challenges I think that's going to be presented to the work group is how do you work with a population that, while generally is fairly sedentary, but when it does move, it can go from a condition of 0 people to 50 people overnight, literally. I'm exaggerating a little bit, but it can change very, very quickly. And how do we try to study this issue? And we're certainly looking for any input from the state of ideas and new approaches, and happy to share as well the successes that we've had on the San Diego River in the city of San Diego. So just wanted to make those comments.
Thank you.
Thanks, Rob.
I would also want to put in, this is Nick Martirano, Director of the Council, to put a plug in for the trash monitoring work group, which Rob sent some staff down to participate in a trash event we hosted in November, which we're very grateful for. But there's also a homelessness subcommittee of that monitoring work group, which is actually meeting in Southern California from 1 to 4 today. But they're looking at homelessness as it relates to trash, but the bacteria impacts are ingrained in the same effort. So I think that there's an opportunity to coordinate there as well.
You'll have enough time to leave this meeting to go there.
I will.
All right, thanks.
Just a procedural question on the work plan. Is it going to the regional board for approval, or is it going to be something approved administratively by the executive officer? Do you know? It's not a big deal.
I think that the work plan, I think the executive officer is the one who's designated to approve it, but I'm not 100% sure on that.
Yeah, no problem.
Yeah.
So when is the deadline to, what are the deadlines associated with the die-off? Going to finish everything in 5 years, or?
June of 2024 is the deadline.
Exactly 5 years.
Yeah, to have it complete.
So So the final report, right?
Mm-hmm. So it's a lot to do in a short amount of time.
Thank you.
Mm-hmm.
Thank you.